Hello everyone, I welcome you all to the 13th lecture of this course. The 13th lecture is on nanotechnology in tissue engineering. So in this lecture we are going to learn what is tissue engineering and why we need tissue engineering and what are the tools required for tissue engineering, and also we are going to learn what are the applications of nanotechnology in tissue engineering. So let us see the definition of tissue engineering. So tissue engineering is an interdisciplinary field that applies the principles of engineering and life science towards the development of biological substitutes that restore, maintain or improve the tissue function. So here why we need tissue engineering? To replace the diseased or damaged tissue, okay, because the donor tissues and organs are in short in supply. We do not have sufficient donors for replacing the damaged tissues or organs, and also if we take the organs from a different person or different animal, our immune system will reject it. So to minimize the immune system response, we can use our own cells and we can engineer the man-made tissues. Okay. So here tissue engineering is an approach to treat patients who need a new organ or tissue with the man-made organ or tissues. So here the tissues are engineered using a combination of patient's own cell and polymer scaffold. So here we are going to take the patient's own cell and we are going to add the cells on the polymeric scaffold, and we are going to grow the tissue in the lab condition and we are going to implant it into the patient. So here the polymeric scaffold will mimic the natural ECM, that is extracellular matrix, okay. So which brings cells together and control the tissue structure and regulate the function of the cells, okay. So here the scaffold act like a support and it will allow the cells to grow, okay. It will provide a support like a extracellular matrix ECM, and also it allows the nutrients to permeate through the porous structure of the extracellular matrix. So this is a overview of tissue engineering. So the first step is we are going to take the cells from the patient's body and we are going to cells in the lab condition, and once we grow the cells, sufficient number of cells, then we can seed the cells on the 3-dimensional scaffold. So then the cells grow on the 3-dimensional scaffold and we can implant the same cells into the repair or damaged site, okay. So we are re-implanting the engineered tissue to the damaged site. So this is the overview of tissue engineering and why we need 3-dimensional, okay. So in case of 2-dimensional, that won't mimic your body condition, okay. So inside our body we have all the organs are in 3-dimensional. So we need a 3-dimensional scaffold, so which mimic like your in vivo condition, and also it act like a support to grow the cells, and which could be useful for making a artificial tissues or organ to replace the damaged tissue or organ. So the tools required for tissue engineering is cells, scaffold and cell signaling. The first thing is cells, so that is the living part of tissue, okay. So that produces a protein or and provides functions of cells, okay. And next one is scaffold, so this provides the structural support and shape to construct, and it should be usually biodegradable and biocompatible. Biocompatible means it should be compatible to the biological system, and biodegradable means if it degrades inside the system, it should not induce any immune response or toxic effects. And third one is cell signaling. So the cell signaling, it may be like a growth factors or hormones, okay. So that is going to tell the cell it has to differentiate or not, what kind of cells it has to differentiate, okay. So these are the three things required for your tissue engineering. The first one is a cell source. It can be your embryonic stem cells or it can be your adult stem cells, okay. And next one is your extracellular matrix, that is your support or scaffold. It can be a metal, ceramics, synthetic polymers and natural polymers. So depends on the tissue you want to grow or depends on the organ you want to grow, you have to select your scaffold. It can be metal for some of the bone replacement, and it can be synthetic polymer and natural polymer. Depends on the application, you have to select the ECM, extracellular matrix. And third one is signals or growth factors. So these growth factors, it is going to decide what kind of cells it is going to differentiate, okay. And also some of the tissues we have to apply mechanical force. This mechanical force also plays a major role in differentiating the cells. So let us see one by one. So what is a stem cell, okay. So it is a cell which has the ability to divide for indefinite periods, and it can be like often throughout the life of the organism, okay. So stem cell is like your in a simple term it can assume like it's a seed. So from the seed you will get the plant, from plant you will get the flower, again you will get the fruit and seed, okay. So similarly the stem cells, it can differentiate into any kind of cells, okay. So under the right condition or given the right signals, the stem cells can given rise to many different types of cells. Okay. So, if you are inducing the stem cells with the suitable growth factors, and it will convert into liver cells, or it can be converted into kidney cells. Okay. So, that is called as differentiation. So, the stem cells again divided into 3 types: totipotent, pluripotent and unipotent. So, totipotent is embryonic stem cells. From embryonic stem cells we can get any kind of cells, and multipotent cells, example is mesenchymal stem cells. From here you can get multiple types of cells, and unipotent is you can get only single type of cell. You can see here the totipotent is an embryonic stem cell, you can get any kind of cells, and pluripotents or multipotent you can get multiple types of cells, and unipotent is your adult liver cell example. Okay. So, you can get the liver stem cell, from the liver stem cell you will get the liver cells like hepatocyte cells. So, it is only one type of cells, we can get it from the unipotent cells. So, we can use the stem cells, and depends on the growth factors, okay, or the hormones whatever you add, the cells will differentiate, the stem cells will differentiate into liver cells, or it can differentiate into kidney cells. So, next one is scaffold. So, the cells are often implanted or seeded into a artificial structure capable of supporting 3-dimensional tissue formation. Okay. So, these structures typically called as scaffold. So, the scaffold should serve at least one of the following purposes. Okay. So, it should allow the cells to attach, okay, and it should deliver and retain the cells and biochemical factors, and it should enable the diffusion of vital cell nutrients, and also it should exert certain mechanical and biological influences to modify the behavior of the cell phase. Okay. So, we can have scaffolds in various structures and materials, depends on the tissue we want to grow, we have to select the suitable scaffold, and the scaffold should encourage the cells to grow, okay, and it should have sufficient porous nature so that it can allow the nutrients to permeate, and it should be highly biocompatible and biodegradable, it should not harm the patient. So, let us see the steps in the tissue engineering. So, first thing is you have to select the suitable cell source, then you have to select the suitable scaffold, then you have to add the cells on the scaffold and grow it. Okay. So, once the cell is ready, then you have to transplant to the human body. Okay. So, this is a biocompatible scaffold, you can add the human cells and the cells multiply on the scaffold, and you can add the growth factors or matrix proteins. Okay. So, then cells secrete growth factors and human matrix proteins, and it became complete human tissue. Okay. So, using the well designed scaffolds and optimized cell growth, so these are the tissues, these were already successfully engineered, okay, skin, bone, cartilage, these are already successfully engineered, but some of the complex organs like liver, heart and lung, these are still under research, okay, because it has a complex metabolic function as well as it require multiple types of cells. Okay. So, that is why it is very difficult to make artificial liver or artificial heart, but still lot of research is going on to develop artificial liver or artificial heart and artificial lungs. So, let us see the example of skin tissue engineering. Okay. So, suppose if we have a wound on your skin, you are losing some of the cells on the skin, and when the skin contract, it forms a scar, but when you replace the damaged cells with your scaffold loaded with the skin cells, it will regenerate the skin and there will not be any scar formation. Suppose you are having a damage or wound on your skin. Okay. So, you are losing this much amount of cells. So, what happens is when the skin contract, there will be formation of scar. So, but when you replace these damaged cells with the scaffold, there will not be any formation of scar. So, let us see how to repair the skin. So, there are two approaches, one is in vitro and other one is in vivo. In the in vitro, you will add the cells and other regulators, okay, to the tissue bilayer and will implant into the host. Okay. And in vivo synthesis, you will put the scaffold into the host and you add the cells on the host and it will be regenerated in the host. Okay. That is called as in vivo synthesis. So, let us see the example, you can see here. So, this is a wound grafted with the scaffold, and if you have the active scaffold, so there is no contraction, it is blocking the contraction and new tissue formed in skin, there is no scar formation. But when you not putting the scaffold, what happens is there will be a formation of scar due to contraction of the skin. So let us see another example. Here we can see here schematic of wound model in adult mouse liver. So here they are making a wound on the adult mouse liver, and they are replacing with the scaffold loaded with the liver cells, and it is healing the wound. Okay, you can see here without scaffold there is no wound healing. With the scaffold it is closing the wound. So let us see why we need nanotechnology in tissue engineering. So you can see here, so when you use a micropore scaffold, the cells are not able to attach properly, and at the same case in the microfiber scaffold also, and but when you use a nanofiber scaffold, the each cells it can easily attach to thousands of nanofibers. So as you know, the size of cell is in the micrometer. Okay, when you use a micropore scaffold or microfiber scaffold, that is not matching with the scale. Okay, but when you use a nanofiber scaffold, a single cell can attach to thousands of nanofibers that will exactly mimic like your extracellular matrix ECM. Okay, so the cells on microfibrous scaffold have a polarized relationship with one side of the cell attached to the scaffold and the other exposed to physiological media. Okay, so in comparison, it is likely that the cells are more naturally constrained by nanofibrous scaffold. So the cell is in the micrometer size. Okay, and when you use the micropore scaffold, what happens is some cells are partially binding to the support and some are exposed to the medium. When you use the microfiber scaffold, again, so when you have the microfiber, the cells are growing on the axis of the fiber. Okay, so again it is not exactly not matching with your ECM. So but when you use the nanofiber, so here the fiber size is in the range of nanometer and your cell is in the range of micrometer. So each cell can bind and attach to thousands of nanofibers. So it exactly mimic like your ECM, extracellular matrix. So that's why we need nanofibrous scaffold for tissue engineering application. So you can see here this is your extracellular matrix. So each tissues or organ we have this kind of extracellular matrix. On the top of the matrix all our cells attach and grow and forms the tissue or organ. So when you use the nanofibers based scaffold, it exactly mimic like your extracellular matrix. Okay, and here you can see here the extracellular fiber matrix, it is in the range of 50 to 500 nanometer diameter. So the cell size is between 10 to 100 micrometer. So the single cell, here you can see here, this is a scanning electron microscope picture. So the single cell can bind to thousands of nanofibers. So the single cell contains thousands of fibers. So the transmission of fine signals can be easily achieved. So what are the techniques available to achieve nanofibers for tissue engineering? First one is self assembly, second one is phase separation, third one is electron spinning. So we will see one by one. The self assembly, which I already explained in one of the lecture, that is protein nanotechnology. So we can use the peptides with hydrophilic head and hydrophobic tail. Okay, so it can self assemble and form the three-dimensional scaffold. So when you use the peptide of 16 amino acids or more and with alternating polar and non-polar, okay, it forms a stable beta strands and beta sheets and forms nanofibers with good hydrophobicity and hydrophilicity. Okay, so using the self assembly we can make the three-dimensional nanofibrous scaffold. And next one is phase separation. So this is a process involves dissolving a polymer in a solvent at high temperature followed by a liquid-liquid or solid-liquid phase separation induced by lowering the solution temperature. Okay, so here we can have a wide range of geometry, dimensions and irregular pore structures, and it is simpler than the self assembly process. So let us see phase separation detail. Okay, so it is a thermally induced phase separation is a technique. Okay, it is mainly useful for generating scaffolds with specific pore size. In this method, the temperature of the polymer solution is adjusted to a point at which a polymer rich and polymer poor phase is generated. Then the solvent is removed and the polymer rich phase solidifies, forming a porous solid structure. which is then freeze dried. Okay, and nanofiber scaffolds with varying fiber diameters and pore size can be generated by adjusting the polymer concentration, the type of solvent, and the phase separation temperature. By simply adjusting the polymer concentration, type of solvent, and phase separation temperature, we can have a various size, porous size nanofibrous scaffold. And here the fibers ranging from 50 to 500 nanometer diameter can be easily achieved, and which will exactly mimic like your extracellular matrix. So this is a phase separation. So the solvent and polymer is mixed and form the solution, and with respect to temperature we can separate the phase, okay, and you can get the different porous and fibrous scaffold. So here the drawbacks are, it is limited to only several polymers and it is only small production scale. So you can see here this is a scanning ultra microscope picture of nanofibrous scaffold with spherical macropores. So the next approach is electrospinning technique. Okay, so this is a process by which high static voltages are used to produce fibers from a polymer solution. Here we can get micron to nanometer size diameter fibers, and fibers have huge surface area to volume ratio. So how to make nanofibers using electrospinning? Let us see a simple example. It is similar to your cotton candy. So in cotton candy, what is your polymer solution? You will be using the sugar solution. Okay, so here when you use the sugar solution and spun out through the tiny holes, as the fiber spins, it is pulled thinner and thinner. Okay, so you will get this kind of fibrous structure. So the simple example is cotton candy. The same principle is for your electrospinning setup also. Okay, so it is a versatile method to produce fibers with diameter in the range of nano, and here we will be having a syringe. Okay, it is connected to high voltage power supply. So when you apply the high voltage between the spinneret and collector, a polymer fluid, okay, slowly pumped out through the spinneret, and the droplet is held by its own surface tension, okay, until it gets electrostatically charged. So after a threshold accumulation of charges, this polymer assumes a conical shape. This is called as Taylor cone, and the thin stream of fiber eludes the droplet, then it forms a nanofiber. So here the electrical field pulls on a droplet of a polymer solution at the tip of the syringe and pulls out smaller liquid fiber, and it is pulled thinner and thinner as it approaches the collection plate. So here we can easily control the dimension of your nanofiber, okay, by simply adjusting the polymer solution viscosity and also the distance between the spinneret and collector. Suppose you are having a more distance between your spinneret needle and collector. So you will get nanofiber. So the distance between this spinneret and collector is very important, and if you reduce this distance, you will get micro size fiber. If the collector is moved closer, okay, you will get micro size fiber, and when you move the collector more far away, what happens is you will get the fiber, you will not get the continuous fiber, you will be having a fiber with break. And again this electrospinning available in vertical as well as horizontal, and the collector can be static collector or it can be rotated drum collector. So using the rotating collector and stationary collector, we can have the aligned and random fibers. So when you use the rotating collector, what happens is it is similar to your thread bundle. Okay, so all the fibers are rolled into the rotating collector and you will get the aligned fibers. But when you use the stationary collector, what happens is you will get this kind of random fibers. So when you see under this scanning ultra microscope, you will get this kind of beautiful aligned fibers when you use the rotating collector, and you get this kind of random fibers when you use the stationary collector. And the main advantage of this nanofiber by electrospinning is it is a simple equipment, okay, and multiple polymers can be combined at monomer level or fiber level and scaffold level. And here we can control the fiber diameter by altering the concentration or viscosity of your polymer, and also the fiber length can be unlimited. So let us see the overview of this nano scaffold and what are the advantages and disadvantages of each method. So self assembly is very difficult to make, but it produces a nanofiber scaffold which closely mimic your extracellular matrix, but the drawbacks are lack of control and limitation of polymers, okay, and also only short fibers can be created and low yield. But when you use a phase separation, it is easier when compared to the self assembly, and here we can make the tailorable and mechanical properties with specific pore size, okay, and here we can maintain batch to batch consistency, but it is limited to lab scale production, and also we can use only several type of polymers, okay, only limitation to particular type of polymers, and electrospinning is very easy and it is cost effective, okay, and we can make a long and continuous fiber, and production of aligned fibers is possible, and we can also tailor the mechanical properties, size and shape, okay, and the limitations are some cases we have to use the organic solvents and we do not have any control over the 3-dimensional pore structure, okay. So these are the advantages and disadvantages of each method for making nanofibers, and whenever you make nanofibers or any nano scaffold, the first thing, important thing is hydrophilicity of your material. When your material is hydrophilic, so it will allow the cells to attach and grow nicely, okay. So how to measure or how to check the hydrophilicity of your material? By using contact angle measurements. So here we have to rest a small drop of water on a solid surface, okay, and a tangential outline of the droplet on the solid forms the contact angle. So here you will be adding a drop of water on the nanofiber scaffold. So when you add a drop of water and using a contact angle measurement setup, we can measure the contact angle, okay. So if your water droplet is giving this kind of contact angle like less than 90 degree, that is your material is hydrophilic, okay. If you are getting a contact angle more than 90, that means it is hydrophobic. If your contact angle is more than 150, it is your material is super hydrophobic, okay. So using this simple experiment, we can understand whether your nanomaterial or nano scaffold is hydrophilic or not. So let us see the applications of nanotechnology in tissue engineering. So using nanotechnology, we can make several nanostructures, nano gratings, nanofibers, nanopits, okay. So the living cells are highly sensitive to local nanoscale topographic patterns within the extracellular matrix, and this cell nanotopography interactions will decide the cell morphology, and also it can induce the differentiation of your cells. If you are adding the same cells on this kind of structures and nano gratings, and it can allow the cells to differentiate, okay. These nano gratings or nanofibers, it can also induce a cellular differentiation and also cellular signaling. So let us see the example of cardiomyocytes. So this is your organ and this is a natural tissue structure, cardiomyocytes, okay. So this is a engineered scaffold. Here the grooved arrays can promote cardiomyocyte elongation and alignment. So we can have this kind of nanostructures. So when you have this kind of nanostructures, the extracellular matrix forces cardiomyocytes to couple mechanically, and this nano grooved surface can force cell alignment in a same way which mimic the natural tissue structure. The next example is epithelial cells, okay. So here the epithelial cells are polarized and adhere to other cells, and when you make the surface, okay, nanofibers modified with surface molecules, so that can promote this kind of natural tissue structure effect, okay. Next example is bone, okay. So the osteoblasts influenced by bone matrix, and we can make the hydroxyapatite nanostructures and with the porous structure similar to your bone, okay. So these nanostructures, it can enhance the osteogenesis, that is formation of bone. The next example is we can have this kind of scaffold with the magnetic nanoparticles and which is carrying the growth factors. When you apply the external magnetic field, it will release the growth factor according to the external magnetic field, okay. So when you apply the external magnetic field and according to the growth factors, the cells will have different kind of forces, mechanical forces. So when you expose to these mechanical forces, this mechanical forces will induce the gene expression and it leads to expression of various proteins, okay. So that is called as mechanotransduction, okay. And we can also tissue engineer the neural cells. So in the nanofibers, we can incorporate the supporting cells and we can also incorporate the growth factor, and which will be useful for neural cell tissue engineering. So let us see how these cartilage cells can be tissue engineered through nanotechnology. So this articular cartilage is a connective tissue that lines the ends of articulating bones, okay. So that provides a frictionless motion and also it is protecting the bones of joints. So we can have this cells and seeded the cells on the scaffold, and when we add the bioactive growth factors and suitable environment, like we have to give the mechanical force, okay, then the nano composite will form the cartilage cells in the artificial scaffold, and that could be injectable or it could be implantable into the patient who has the damaged articular cartilage tissue. Okay. So next one is we can also engineer the bone cells. Here the bone is a specialized form of connective tissue that forms the skeleton of the body. Okay. So mimicking the bone structure and its properties present an important frontier in the fields of nanotechnology and material science. So using with the help of materials and nanotechnology, we can engineer the bone. Okay. So here we can have the synthetic biomaterials made up of hydroxyapatite and collagen, which mimic like your bone scaffold. Okay. And we can add the bone marrow stem cells, and which can be regenerate your bone, damaged bone. So let us see vascular cells tissue engineering through nanotechnology. So here we can harvest the cells from the patient and grow the cells in the lab condition. Okay. And at the same time you can harvest the arteries from the pig, okay, and do the decellulating process. It will remove all the cells, and we can use that arterial matrices. On the top of that you add the cells which we isolated from the human, okay, that means recellularizing the porcine matrix with the patient cells, and when it is matured and it will grow and it form the functional vascular graft. So here we are using the arteries from the pig and the cells from the human, and we are making the functional vascular graft. So the next example is hepatic cells tissue engineering through nanotechnology. With the help of micro nanotechnology, we can make the micro nanostructures which mimic like your liver, okay, and when we add the hepatocyte cells, and it will grow like a complete liver. And next one is we can also use it for dental tissue engineering. Okay. So here we can take the dental pulp stem cells and add it on the scaffold. Here the scaffold can be collagen or hyaluronic acid, okay, and once the cells grows on that, we can implant into the alveolar bone, okay, and dental pulp. So it will regrow the dental tissue. So the next application is nano devices in tissue engineering. So here 3-dimensional freestanding nanowire transistor probe for electrical recording. So this probe is composed of a nanowire, this is a nanowire, okay, and a flexible substrate material, and this device is used to penetrate the membrane of living cells and measure the intracellular signals. You can see here this small nano device can penetrate the cells and it can measure the intracellular signals. So the tissue engineering is not only useful for making the artificial tissues to replace the damaged tissues or damaged organ, it could be also useful for making some edible tissues, that is test tube based burger. Okay. So here you can see the example, the tissues are taken from the animal muscle and the stem cells are extracted from the tissue, okay, and the muscle cells are grown under the tension to bulk them up, and the new muscle fibers are minced and turned into burgers. Okay. So this will avoid the slaughterhouse and killing of lot of animals. Okay. So we can have a test tube grown burger. So let us see another example from pig to plate. Okay. So this is an article published in Nature. Okay. So here the researchers are adapting tissue engineering techniques to grow edible meat. Okay. So the tissue engineering is not only for making artificial tissues for damaged organ or biomedical application, we can also make the edible meat. Okay. So take a small biopsy from the pig and grow the cells and add animal free growth serum to multiply the cells, okay, and also give some mechanical force so that it can exactly mimic like your muscle cell. Okay. Then you can grind up the muscle strips and you can add flavor like iron or vitamins. Okay. So that will add flavor to your meat. Okay. And finally you can cook and eat. That means from this example we learnt like the tissue engineering is not only for biomedical application, it can be also useful for growing the edible meat. Okay. So as a summary of this lecture, okay, so in this lecture we have learned what is tissue engineering and why we need tissue engineering, and also we have learned what are the tools required for tissue engineering, and also we have learned what are the various applications of nanotechnology in tissue engineering. I will end my lecture here. I thank you all for listening to this lecture. I will see you in another interesting lecture.